Good day. This is our GMO proposal for today. Your attention will be highly appreciated.
Before we start, first, what is GMO? Well, GMO stands for Genetically Modified Organisms or GMOs. This can be defined as a life form. Examples are plants, animals, or microorganisms, in which the genetic material DNA has been altered in a way that does not occur naturally by mating or natural recombination.
Now that we know what GMO is, we can finally tackle our GMO proposal that will benefit us and help for others.
Rice. What is rice? Rice is a sustenance that we consume every day that has many nutritional values. Rice is also the main source of food for more than half the world's population, including 640 million undernourished people living in Asia. Rice includes more than 15 essential vitamins and minerals, including folic acid, B vitamins, potassium, magnesium, selenium, fiber, iron, and zinc. But rice does not contain vitamin C. It is important to note that the nutritional profile of rice can differ based on how refined it is and whether it has been enriched with vitamins and minerals.
The supply will increase and the price of rice will lower as time goes on, thus making it accessible for people in poverty. There will also be no use of pesticides and other pest killers, thus making the sustainability of it sustainable for at least 10 years. The resources needed is also manageable. In our research, we found that one of the common ways to modify a crop is through selective breeding through many generations of crops. So, we can say it is indeed possible to try.
In addition to vitamin C and folate, oranges provide smaller amount of nutrients, including calcium, potassium, and thiamine, or vitamin B1. Oranges are a good source of fiber and rich source of vitamin C and folate, among many other beneficial nutrients. The vitamin C in oranges help your body in a lot of ways, such as 60 calories, no fat or sodium, 3 grams of fiber, 12 grams of sugar, 1 gram of protein, 14 microgram of vitamin A, 70 milligram of vitamin C, 6% of your daily recommended amount of calcium, 237 milligrams of potassium, 15.4 grams of carbohydrates. It also protects your cells from damages and help your body make collagen and protein that heal wound and give you smoother skin. Make it easier to absorb iron to fight anemia, boost your immune system, your body's defense against germs, slow the advance of age-related macular degeneration or AMD, a leading cause of vision loss, helps fight cancer-causing free radicals.
And now, let's see what vitamin C is. Vitamin C is known to be an antioxidant that helps protect your cells against the effect of free radicals molecules produced when your body dissolves food or exposed to tobacco smoke and radiation from the sun, X-rays, or other sources. Free radicals might play a role in heart disease, cancer, and other diseases. Oranges are one example of an efficient source of vitamin C.
Since people around the world consume 100 kilograms of rice per person every year, what if we were to combine rice and oranges' nutritional value and other benefits together?
Great question, Dutch. Well, if we were to combine rice and oranges, there will be a massive change. Like so, more people will be able to consume rice daily than before. There will be a built-in flavor or the sweetness, more added sustenance in the rice. It will help farmers lessen the use of insecticides, has a longer shelf life, and higher yield and faster production. But how will we achieve this?
The process of this will not be easy and will require a lot of time and hard work. The genes of oranges will be inserted into the genome of rice in a way to improve it. This way, rice can and will offer more nutritional value and added a sweetened flavor.
Like other GMO crops, we will also add a gene from a bacterium called Bacillus thuringiensis, which will cause the cells of the rice plant to produce insecticides in their leaves that will kill insects and pests, leaving the crop undamaged. It will result in a massive boost of rice production, providing more rice than before. With the Bacillus thuringiensis inside the crops, farmers won't have to pitch in buying insecticides, thus saving them time and money.
Here are the steps and process of our project. Step 1. Identify a trait of interest, which is the rice and oranges vitamin C. Step 2. Isolate the genetic trait of interest. Step 3. Insert the designed genetic trait into a new genome with the use of restriction enzyme. The gene to be transferred must be cut out of the originating organism and separated. Restriction enzyme which function similarly to molecular scissor recognize particular sequence in DNA and cut it at those locations. Since restriction enzyme is only applicable to non-methylated DNAs, the orange's source of DNA of our project should be far from right.
An enzyme called a restriction endonuclease cuts DNA strands at a precise spot. It searches the DNA for a specific target sequence and then cleaves the DNA when it finds it. The target sequences aren't very long. The common restriction enzyme, ECOR1, for example, only has a 6 base pair target sequence. Thousands of restriction endonucleases, or RE, have been isolated to date, the majority of which are from bacteria. Because these enzymes can recognize and cut foreign or viral DNA, bacteria use them as a defense mechanism.
And the nucleus that cuts double-stranded DNA in a variety of ways are known as restriction endonuclease. It sometimes cuts both strands at the same time, resulting in blunt ends. Occasionally, it cuts each strand at a different place, resulting in overhangs. Overhanging means one strand is longer than the other, and this is sometimes referred to as sticky ends.
To insert the DNA, we're going to use the gene gun method. Gene gun is the technique of biotechnology usually used in plant biotechnology. It is one of the methods of genetic engineering. It is also called as bialistic method. It is commonly used to insert genetic material into the cells by the particles coated into small DNA sequences.
Gene gun bombardment is a method of the physical introduction of DNA into plant cells containing cell walls. The gene gun is utilized to bombard the plant cell wall with many DNA-coated metal particles by using compressed helium as the propellant.
The basic steps to use the gene gun are: 1. Prepare the tungsten particles and coat them with DNA. 2. Load the particles onto macrocarriers. 3. Prepare the target cells. 4. Decide on your parameters and set the gene gun accordingly for distance from target, pulse duration, and CO2 pressure. 5. Place the target under the gene gun barrel and fire it. Step 4. Growing the GMO. To produce a GM plant, new DNA is transferred into the plant cell. Usually, the cells are grown in tissue culture where they develop into plants. The seed produced by the modified rice will inherit the new DNA.
And that is all. Thank you for listening. We hope you enjoy our presentation and hope that we explained everything clearly.